know, if you see the structure of an animal cell, how the actually animal cell looks like, and who were the scientists actually who try to give a detailed account of the animal cell, not only the animal cell but cell in general. Who were the scientists actually who contributed to the structure of a cell, and also who invented various microscopes, and based on their opinions, whatever the rules they have given, we have already seen. So if you look at the history, already we know that actually the cell is nothing but it means a very, very small room. So it is a basic unit. Actually, it has been described by various scientists as the basic structural, functional, as well as a biological unit of almost all the living organisms. So the cell plays a very, very important role. And we have discussed that the origin of cell actually on this particular earth has started or has begun at least 3.5 billion years ago, and it is the smallest unit of life. It is a biological unit of life, functional unit, structural unit. So it is very, very important for the living organisms. So the study of cells, as we all know, you can call it as a cell biology or the cellular biology, or you can also call it as a cytology. And the structure unit of the cell, since it is called as a cell, so it can also be called as a unit of life, because the cell performs various functions of the body which are basically required for the functioning of life. So that is why we can also call it as a unit of life. And not only the cell has been identified, but several authors actually also have given various laws or various theories, like cell theory and all those things. So the cell theory says that the cell is the basic unit of life. And several authors actually have contributed to the discoveries. Most of them have, discovered the microscopes, and then they, based on their opinions, they have actually given their views regarding the structure of the cell. So 1665, the cell was first discovered by Robert Hooke. Then Anton van Leeuwenhoek discovered the protozoa, and he saw the bacteria. And later on, Robert Brown discovered the cell nucleus. And according to the Shieldon and Swan, who has actually given the cell theory, according to them, the cell actually is considered as a unit of structural and functional unit, as well as a biological life of an organism. Then next, we have also talked about the this thing, like Shieldin and Schwann just have told, they have proposed the cell theory, and von Rolliker realized that sperm cells and also the egg cells are also the cells. Now, 1856 actually, the scientist by name Prinzhorn observed how a sperm cell will penetrate the egg. Then Rudolf Virchow in the year 1858, what he did actually, he said that the cells develop only from the existing ones. That is, the cells actually will come from the pre-existing ones because of cell division. Then 1857, Colliker described the mitochondria. Then 1879, Fleming described about the chromosome behavior during mitosis. In 1898, Camillo Golgi actually is called as described the Golgi apparatus. In 1938, then Behrens used differential centrifugation to separate the nuclei from the cytoplasm. So like this, different scientists actually have described the cell in various ways based on their discoveries. Now coming to the introduction, you all know. So it is a basic as well as a structural and functional unit of all living organisms, and it contains hereditary information, so which is passed from one cell to another cell during the cell division. And all living organisms, you all know, they're made up of cells. So because the cell is a basic unit, the cells will form the tissue will make up an organ, and the organ actually will make up a full-fledged organism. So like this, the cell is considered to be very, very important. Now, some of these living cells, actually, if you talk about based on the number of cells, some organisms consist of only one cell, some organisms contain a number of cells. So based on their the number of cells, you can call them a single cell, that is unicellular. And if they possess more than one cell, you can call them as multicellular. And the cells that constitute an animal or any living organism, you call as an animal cell. So you all know actually all the living organisms like plants, animals, human beings. So they are all living organisms, and these living organisms actually contain the cell. And when you see a difference between the plant cell to that of an animal cell, like prokaryotic, first before going to that, we can go to prokaryotic as well as eukaryotic cell. Prokaryotic actually is a primitive cell which lacks the nuclear membrane, the new plasma membrane, as well as the various organelles. But a eukaryotic cell, like you have plants, animals, as well as a human being, they consist of not only the plasma membrane, but they consist of nucleus, genetic material, as well as all the different organs. So the number of cells again also varies. Now if you compare, if you differentiate the plant cell as well as the animal cell actually is a eukaryotic cell, no doubt. Even plant cell is a eukaryotic, but in the animal cell you have got a plasma membrane, whereas in a plant cell you have a cell wall. And the number of cells in the animals also will vary from organism to organism. Not only the number of cells vary, but also the shape of the cell as well as the size of the cell also will vary. They say that about human beings contain somewhere about 40 trillion cells, whereas the human brain consists of around 80 billion of these cells. And shape of the cells also varies. It may be flattened, cubical, columnar, fusiform, stellate, pyramidal, flash-shaped, and so on. Eukaryotic cell, if you talk about, it is very, very microscopic, and the size also will vary from 1 micron to about 275, 75. They say that the longest cell actually is the human nerve cell, and the largest cell actually is found in the egg of ostrich. And coming to the what do you say, the eukaryotic cell, just now I have already talked to you. That is about the animal cell. Animal cell consists of the plasma membrane, and it also consists of the protoplasm. Protoplasm is nothing but it is actually a combination of cytoplasm plus the nucleus, where cytoplasm actually consists of all the organelles, whereas the nucleus consists of the nuclear membrane along with the nucleolus as well as the genetic material. So different types of microscopes, as we have already discussed that without the invention of the microscope, the structure of the cell would not have been possible by various authors, because cell is very, very minute. So the structure of a cell can be visualized only under the microscope. So without the microscope, the study of cell is highly impossible. So in those days, actually, various scientists have developed these microscopes. But the simple microscope came, then the compound microscope came, then later on about the electron microscope. Now, Robert Hooke was the first scientist actually who tried to develop the simple microscope. Then came the compound microscope. So like this, different types, and even the dissection microscope. But these types of microscopes gave only a clear cut, only a basic idea about the structure of a cell, because the internal organs, that is what we are talking about, the different organelles that are present in the cytoplasm was could not be visualized under the ordinary microscopes. Because here in this case, in the ordinary microscopes, if you observe, the source of illumination is light, though it may be an artificial light or it may be a natural light, but the magnification is very, very less. So that is the reason why minute details could not be observed by using these microscopes. But when the electron microscope has been invented, the electron microscope's dissolving power as well as the magnifying power is very, very more when compared to that of these, because there the illuminating source is electrons, and electrons have got high resolving power, and also they have got high magnification. So that is the reason why these electrons actually which are being used to generate the the what do you say, the current or anything, in case of this electron microscope, has helped the scientists to observe very keenly even the minute details. So with the invention of electron microscope only, the scientists, especially the cell biologists, they used to give, they gave us a very detailed structure about the cell, whether it may be a plant cell or an animal cell or a what do you say, the human cell. So that is how the different what do you say, the structure, you can say, of the cell, minute structure of a cell could be observed. This is the flow chart already we have discussed. The animal cell, if you take, it is bounded by means of a plasma membrane. Then it has this protoplasm. Now, beneath this plasma membrane, you have got the protoplasm, which is again differentiated into cytoplasm as well as the nucleus. Cytoplasm consists of again paraplasm as well as the cell organelles. Paraplasm actually consists of nothing but the inorganic substances, like in the form of water, organic as well as inorganic components also, like carbohydrates, proteins, fats, enzymes, salts, as well as the other substances. Now, apart from these, it also has got the cell organelles. Now, if you see a cell, actually, the 70 to 75% of these cells is made up of nothing but the water, whereas the remaining 30% consists of nothing but the cells. Okay, so now coming to the cytoplasm. Cytoplasm consists of, apart from the paraplasm, it also have got various cell organelles, and these cell organelles again play a very, very important role. Each organelle actually is unique, and each organelle plays a very significant role in the metabolism of the cell. So what are those? That is endoplasmic reticulum, mitochondria, ribosomes, Golgi apparatus, lysosomes, then centrioles, mitochondria, as well as the basal granule. So these are some of the structures, or you can say the organelles that are being present in an animal cell. Now coming to The ultrastructure of the animal cell. Last class I have already have told you. So different shapes as well as the sizes of various cells like sperm cell, nerve cell, muscle cells, and you have got the liver cell. And ostrich egg is very very big. You all know this is the basic structure. This is the basic structure of the animal cell. Now, this animal cell actually, if you see, it is bounded by means of a plasma membrane, and beneath this plasma membrane, you have got what? You have got the cytoplasm as well as the nucleus. This pink colored actually is nothing but the nucleus, and you have yellow color. I mean, cream color is the cytoplasm. Again, in the cytoplasm, you have got different organelles are there. Okay, and in the nucleus, you have got the nucleolus as well as the chromatin network or the chromatin reticulum. Then different structure, I mean the structure of different organelles we have seen. First, we have talked about the plasma membrane. You all know it is a membrane which bounds the cell. It is a thin elastic membrane and it is bounded by a double unit membrane, and it is uniform in thickness, and it is made up of lipoproteins, not only lipoproteins, but it also consists of the proteins. So they say that each unit membrane actually is made up of a bilayer of phospholipid along with I mean, between an outer as well as an inner membrane protein layers. And the term plasma membrane actually was first coined by Nageli, and this plasma membrane actually breaks and rejoin again to form the whole structure is called as fluidity, because whatever the content, that means the lipids, the proteins, as well as the carbohydrates, as well as the enzymes, that is actually present in the plasma membrane. They are present in a semi-solid state. They are present actually in a fluid state or a quasi state. So that is the reason why actually the plasma membrane is more flexible. It is more, more flexible, and it can break and it can again rejoin. So that is why this fluidity actually helps the plasma membrane, not only the plasma, that is the whole cell as such, to undergo various processes. So the fluidity or the fluid nature of the plasma membrane actually is very, very important, as it plays a very, very important role in performing the various, the transport activities. Now, the functions, if you see, the plasma membrane gives rigidity to the cell, stability to the cell, maintain the structure of the cell, and it also regulates the flow of various ions, molecules of various substances. And for these different methods, transport methods actually are being employed in the form of osmosis, diffusion, endocytosis, exocytosis, as well as a phagocytosis. So these are about the plasma membrane. So this is a diagram of the plasma membrane. Already we have discussed. Okay, now we'll go to the other one. Then next we have talked about the cytoplasm. Just beneath the cytoplasm, plasma membrane, you have got the cytoplasm, which consists of what we can also call it as a cytoplasmic matrix, or the kinoplasm, or the hyaloplasm, or the cytosol. And it is colloidal in nature, transparent, colorless, homogeneous fluid, and it is actually present between the nucleus as well as the plasma membrane. And this cytoplasm, it consists of two parts. One we call it as organelle, and the other one we call it as the paraplasma, or the inflorescence, or the non-living structure. The paraplasma or the inflorescence of the non-living structure are nothing but your glycogens, fat globules, proteins, enzymes, inorganic substances, as well as a pigment. Whereas the various cell organelles that are present are mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, peroxisomes, phagosomes, filament, fibrils, centrioles, as well as the nucleus. So they perform various activities. So the main function of the cytoplasm actually is nothing but it is involved in secretion, respiration, protein synthesis, and all those things. So this is about the cytoplasm. Now we will talk one by one about the structure of each organelle, which plays a very, very important role. First, already this we have discussed about the endoplasmic reticulum, which is called the benchwork of the cell or the cytoskeleton, because it gives a framework, it gives a skeleton, and actually it is being present in the form of a network. So that is the reason why actually it gives a benchwork of the cell or the cytoskeleton. It was first reported by Porter in the year 1953. Again, it also has a membrane-bound organelle. It has two types observed in the cytoplasm. I mean, two types of endoplasmic reticulum. One is called smooth, and the other one we call it as a rough. And a smooth endoplasmic reticulum do not contain any ribosomes, whereas rough endoplasmic reticulum actually has got the ribosomes. Now, functions of these are it helps in synthesis and storage of lipid and glycogen, and also detoxification of drugs and poisons. Now, since the rough endoplasmic reticulum has got bounded ribosomes actually it helps in the protein synthesis. So RER is in fact an extension of the outer nuclear membrane. Functions is it helps in protein synthesis. Then diagram of this is a diagram of endoplasmic reticulum. Already we have seen this is yellow and pink part is an endoplasmic reticulum. So the extension of that nuclear membrane is nothing but forms the endoplasmic reticulum. We have two types. One is rough and smooth. Rough has got ribosomes, and smooth does not have got the do not contain any ribosomes. ribosomes. The next we have talked about ribosomes. Ribosome, the term ribosome actually was coined by Pellet in the year 1995. It is described as the engine of the cell, or the RNA granules and also the protein factories. So we can call it as the engine of the cell or the RNA granules as well as the protein factories. Why? Because actually, since it has got the helps in the protein synthesis. Because ribosomes contain the ribosomal RNA, so that is it helps in the protein synthesis. And shape of this, it is spherical without the membrane, and each cell actually contains thousands of the ribosomes, which are scattered freely in the cytoplasm, and they are also present on the surface of endoplasmic reticulum. And each ribosome, if you see, it consists of 60% of RNA and 40% of the protein. And ribosomal proteins, they act as enzymes and also they catalyze the protein synthesis. And each ribosome, you see, it has got two subunits. One is called larger subunit, and the other one you call it as a smaller subunit. Larger subunit actually bound the transfer RNA, and the smaller subunit will bound the messenger RNA molecule. Larger subunit consists of 60S, and whereas smaller subunit has a value of 40S. The main function is nothing but protein synthesis. The next, this is a diagram of the ribosome. We have the larger subunit as well as the smaller subunit. If you look at it over here, this is the larger subunit. It is bound to the transfer RNA, whereas smaller subunit to ribosomal RNA. Okay. So this is the basic structure of the ribosome. Next, mitochondria. Now, coming to mitochondria, again, mitochondria is also plays a very important role. The term mitochondria actually was given by Brenda, and these are actually described as powerhouse of the cell because they store the energy. Okay, so that is the reason because energy is required for the various metabolic activities of the cell. So whatever the energy that is actually being produced is stored in the mitochondria. So that is why it is considered to be the powerhouse of the cell or the storehouse of the cellular energy. And mitochondria, as other organelles, it is also present in the cytoplasm with rod-shaped or oval or spherically shaped. And it has a size of about 0.5 to 10 micrometer. And the number of mitochondria again in each cell varies. For example, mature erythrocytes, they do not have mitochondria, while the liver and muscle cells have got thousands of mitochondria. And similarly, it is bounded by a membrane, whereas ribosomes is not bounded by a membrane. But mitochondria actually is bounded by a double unit membrane called as the outer membrane as well as the inner membrane. And in between these two outer and inner membrane, you have got a space which is filled with the matrix, which is called mitochondrial matrix. Then you have got the cristae, F1 and F2, F0 particle, and various enzymes, ions, and circular DNA as well as ribosomes. Now, the inner membrane of the mitochondria actually basically they fold or they bend into the matrix and actually give an appearance of finger-like projections. These finger-like projections are nothing but called as cristae. And the gel matrix actually is the central mass of the mitochondria. Mitochondria to the presence of circular DNA. So this matrix actually consists of circular DNA, ribosomes, as well as various enzymes. And outer and inner membrane of the mitochondria actually has got the electron transport particles, which are in the form of F1 and F0. So this is about the mitochondria. I think this also we have discussed last time. So this is ultrastructure, functions. If you talk about, it participates in cellular respiration and produces or generate the energy in the form of ATP and through oxidative phosphorylation. And mitochondria also stores calcium and it also helps in cell signaling activity, generating cellular as well as a mechanical heat and also mediating cellular, the growth as well as the death. So this is the structure of the, the mitochondria. Outer membrane, inner membrane foldings are called as nothing but the cristae. So in the matrix, you have got the ribosomes is there like this. The next is your lysosome. Now, lysosome actually they are called as a suicidal bag. Now, the suicidal bag is nothing but they undergo phagocytosis. It means they try to engulf the foreign objects. So it is also called as a suicidal bag, and it was first described by Christian D. Dubé in the year 1955. So here lysosome is bounded by a single layer. Whereas we have seen the mitochondria, which is bounded by double layer, but lysosome actually is bounded by a single layer membrane, and it is spherical or rounded in shape. Now, it exhibits polymorphism in their morphology. It exhibits polymorphism means it occurs in various forms, like they are called as primary lysosome, secondary lysosome, residual bodies, as well as the autophagic vacuoles. So the lysosomes also play a very, very important role. So it contains hydrolytic enzymes. So with the help of these hydrolytic enzymes, what it will do, it will try to phagocytize what do you say, or engulf the foreign object. So that is why it is also considered to be the suicidal bag. But it occurs in polymorphic form. That is, it occurs in the form of primary, secondary, residual bodies as well as autophagic vacuole. Now, function is it helps in intracellular digestion, means extracellular digestion, intracellular digestion, as well as the autolysis. So this is about the lysosome. So lysosome actually contains a lytic enzyme or the hydrolytic enzyme. So because of the presence of enzyme only, it will digest, or it will try to absorb the foreign part or the foreign body. Okay. The next is what? Golgi apparatus or the Golgi complex. Now, the Golgi apparatus or the Golgi complex also plays a very, very important role. It was first discovered by Camillo Golgi in the year 1898. Now, it is also called as lipochondriosome or idiosome. In plants, actually, they call it as dictyosomes. Now, dictyosomes in the sense nothing but a group of what is ribosomes. You call it as nothing but the dictyosomes. So, you can also call the Golgi apparatus or the Golgi complex as the lipochondriosome or the idiosome. But in plants, they call it as a dictysome. Now, the shape of this varies from one cell to another cell. Again, it is a unit membrane and has got 3 layers. So, it consists of 3 layers, which is again a unit membrane. And you have an inner lipid layer and outer 2 protein layers, just like plasma membrane. Now, plasma membrane actually it is bounded by 2 layers, we say, but the inner one, that is a lipid layer, actually occurs in a bilayer form. Whereas the outer as well as the inner actually consists of the protein. So here also, actually, same way, it is also unit membrane, represents a unit membrane, and it consists of 3 layers. That is, it has got an inner lipid layer and the 2 protein layers. Like it is basically, they call it as PLP layer, means outer protein, then middle lipid, and then you have got the inner protein. So that is what we call it as the unit membrane. Now, Golgi appears 3 major components such as flattened cisternae, then you have the large vacuoles as well as vesicles. Means here the Golgi apparatus, or you can say the Golgi complex, appears or occurs in 3 forms. What are those? One is called as a flattened cistern in the form of flattened cisternae. Then you have large vacuoles as well as the vesicles. Now, if you see over here the structure, now see here, this is, so they occur as, they occur as the vacuole, they occur as cisternae as well as secretory vesicles. So that is what. So you may say that Golgi apparatus occurs in 3 forms. One is cisternae, then you have got the vacuoles as well as the vesicles. Now, functions is several functions actually are assigned to this Golgi apparatus. That is, they store, they secrete, and also they form the zymogen granules. That is the secretory granules or the secretory vesicles. And also they produce the lysosomes, pigmentation, as well as the acrosomes. So the Golgi apparatus, the main function actually Golgi apparatus is, you might have heard, if you see the structure of a sperm, on the head of the sperm, near the head, you have got an acrosome. Through this acrosome only, actually, what happens is the egg, or you can say the sperm, will be able to penetrate the egg. So that acrosome formation, or you can say, is nothing but being formed by this Golgi apparatus. So the function of Golgi apparatus is very, very important. So it has got a number of functions, like it can store, it can secrete, and it also can form the zymogen granules, it can form the lysosomes, then it can form pigments as well as the chromosomes. So this is about the Golgi apparatus or the Golgi complex. The next is what? Centrioles. Now, if you talk about the centrioles, now centrioles is also called as the cell center. Now, just below the nucleus, you have got certain structures which are called as the cell centers. Now, these centrioles, you don't find it in the plant cell. These centrioles, you find it only in the animal cell. And this actually has been discovered by von Brenden in the year 1887. Now, von Brenden actually was a scientist who tried to discover the centrioles. Now, and it is located just near the nucleus. Just I have told you, just near to the nucleus, below the nucleus, you have got a hollow cylinder without any limiting membrane. So that is what we call it as nothing but the centriole. So it is located near the nucleus and hollow cylinder without any limiting membrane. But it has got 2 proteins. One is called as the tubulin, and the other one you call it as the dynein. Because these centrioles, the protein which is present in this centriole, that is tubulin, actually helps in the spindle formation during cell division. So that is the reason why centrioles play a very, very significant role. So what are the 2 proteins that are being present? One is called as the tubulin, and the other one you call it as nothing but the dynein. So it has got 2 proteins. Now function is, it has formed asters and spindles of microtubules during late prophase or early metaphase of cell division. So that means it helps in the formation of asters. That's why I told you, it also helps in the formation of spindle fiber, that is spindles of microtubules. Now, the spindle tubules or the fibers actually are separation of the chromosomes, and they bring to the polar regions in the later stage of cell division. Now, the spindle formation of the spindle tubules actually we observe during the cell division, and it is nothing but basically the separation of the chromosomes. And the separation of the chromosomes, you see it during the cell division, that is during the later stages, where they are actually distributed to the two different regions, that is the two polar regions of the cell division, that is during the cell division. So that is why even centrioles has got a very, very significant role to play. Now, next is what? Microtubules. Now, the microtubules, just now I have told you, now microtubules, apart from the centrioles and all those things, the animal cell also possesses a number of these tubules, that is what we call it as microtubules, which are hollow cylindrical tubules. Now, with a length of 200 to 300 angstroms and thickness of about 50 to 70 angstroms. And it is made up of a protein which is called tubulin protein, and it consists of about 13 subunits of microtubules, tubules, which is called as protofilament. Now, each protofilament is globular in shape and has a diameter of about 50 to 70 angstroms. So that is the main, the role of microtubules. So microtubules actually consist of several tubules, and the main important part is it consists of a tubulin protein consisting of about 13 subunits and is called as a protofilament. And each protofilament is globular and has a diameter of so and so. Now, the functions is it acts as the basic unit for the origin of centrioles and kinetosomes, cilia, as well as the flagella. So, actually, from this microtubules only, or you can say, it gives rise to the centrioles, to the kinetosomes, to the cilia, as well as the flagella, which we specifically find in the case of the animal cell. Now, the microtubules are also concerned with the formation of the asters as well as spindle fibers during the cell division. So just now I have talked about the spindle, the centrioles as well as these microtubules. Both of them actually help in the formation of the spindle fibers as well as the asters during the cell division. And also the shape, it gives shape to the cells and also access channels for the intracellular transport of the water molecules as well as the ions. So this is all about the microtubules. So microtubules, like the centrioles, also play a very, very significant role. So it acts as the basic unit. Then for the origin, or you can say for the formation of the centriole, kinetosome, cilia, as well as flagella, and it is concerned with the formation of the asters as well as the spindle fibers during the cell division. It also gives shape to the cells and also act as a channel for the intracellular transport of the water as well as the ions. Now, apart from these, you also have another structure which is present in the animal cell, that is what we call kinetosomes. Now, the kinetosomes are basically called as the basal granules, and they are described as modified centrioles. You can also call them as modified centrioles. And each kinetosome actually has 9 triplets of hollow microtubules, each of which consists of 3 subunits or the subfibers. So each kinetosome, if you observe, it has got 9 triplets of hollow microtubules, and each one consisting of 3 subunits. The kinetosomes are described as the roots of cilia as well as flagella. So the kinetosomes, we can call it as they are called as, or they're described as the roots of cilia as well as the flagella. Okay. So this is about the kinetosomes. The next you have got the nucleus, which is very, very important again. Now the nucleus is placed in the center. Actually, it was first observed by Robert Brown in the year 1831. It is actually described as a dynamic center of the cell, and it is present at the center of the cell, and it comprises of means, or you can say it is bounded by means of the nuclear membrane. And this nuclear membrane has got the nuclear pores, and also the cytoplasm, or you can, which is present inside this nucleus, you call it as a nucleoplasm. And also it has got the chromatin network or the chromatin reticulum with the genetic material as well as the proteins as well as the nucleus. So these are the the components which are present in the nucleus. So it is bounded by a nuclear membrane. It has got nuclear core. It has inside you have got the nucleoplasm with the chromatin and network possessing the genetic material as well as proteins and nucleus. Now they say that it occurs 10 to 15% of the cell volume. I mean, it occupies that much of the volume, and it is spherical in shape. Again, double layered mode of lipoprotein surrounded by a nuclear membrane separating it from the cytoplasm. So in between these two layers, again, you have got a perinuclear space. Perinuclear space. So the nuclear membrane of the nucleus is about 3,000 to 4,000 nuclear pores. So where are the pores located? The nuclear pore, they are actually present on the nuclear membrane, and the size of the pore again varies. It may be 80 mm in diameter, and the nuclear pore actually is funnel shaped. Because you all know actually whatever the genetic information, whatever the transport in the form of water or ions that has to be carried from cell to another cell, or from the cytoplasm to the what is a nucleus, or from the nucleus to the cytoplasm, actually occurs through the nuclear pore, because pores actually help in the passing of these or in the transport of these material. Now, the nucleoplasm of the nucleus actually is a heterogeneous mixture because it contains histones, it contains non-histones, and a number of ions of calcium, sodium, potassium, many enzymes, chromatin, as well as the nucleus. And this chromatin actually appeared network. You all know actually only during the cell division, the chromosomes will attain the shape. But in the ordinary time, if you observe under a microscope, the chromosome actually is present in the form of a reticulum. We can say that is what we call it as chromatin reticulum. Because it doesn't have a defined shape as such or a defined structure as such. So that is why it always appears in a network. So the chromatin appears in a network, and it is like to elongated. I mean, it is an elongated thread-like structure, you can see. So that is the reason why only at the time of cell division we call them as chromosomes. But in the ordinary time, we call them as a chromatin network or the chromatin reticulum. Now, the chromosomes contain hereditary material, that is the DNA and many nucleoproteins. So what does these chromosomes contain? It contains a hereditary material that is in the form of DNA and also many nucleoproteins. Okay. Then you have, so this is the structure of a nucleus. You have got the nuclear membrane. Inside you see this is the nucleolus. This is the nucleoplasm. These are the, okay. And these are the, you have the proteins. You have this in the form of chromatin, what do you say, reticulum. Okay. And these are the nucleopores, what you are seeing. And below this, you have got this rough endoplasmic reticulum. Okay. So the nucleolus actually was first discovered by Robert Fontana. It was first discovered by Robert Fontana. And this nucleolus actually is very, very tiny or a small body, which is a prominent structure which is present within the nucleus, non-dividing nucleus of the cell. And it has a very rich RNA also, and the protein and origin of ribosomes is also being present. So we can say that actually the nucleoplasm contains RNA, proteins, as well as the ribosomes. The main function of nucleolus is nothing but to produce and assemble the subunits which form the ribosome. So whatever the subunits, two subunits we know, no, one is 60S and the other one is 40S. So the main function of this nucleolus is nothing but to produce and also to assemble the subunit to form the ribosome. Okay. Now coming to functions, general functions of the nucleus, it controls the hereditary characteristics of an organism. It produces ribosomes, stores hereditary material, as well as the stores hereditary material, proteins, and is also responsible for the protein synthesis. So this is about the nucleus. If you look at the summary of all these things, so we have seen the ultrastructure of an animal cell, how the animal cell look like, what are the various, what do you say, the the organelles that are present in a cell from the beginning. So basically, in general, we can talk about when we talk about the structure of an animal cell. So ultrastructure of an animal cell, if you look at animal cell, actually is nothing but it is bonded by a plasma membrane. Within the plasma membrane, you have got what the protoplasm. The protoplasm again differentiated into what? Cytoplasm as well as the nucleus. Cytoplasm consists of paraplasm as well as the different organelles in the form of mitochondria, Golgi bodies, endoplasmic reticulum, just now what we have seen. And in the center, you have got a conspicuous nucleus, which is bonded by a nuclear membrane. Within the nucleus, you have got the nucleoplasm and you have got the nucleolus. And apart from that, it also has got the proteins, ribosomes. So that is a brief summary. So this is the name of the organelle. So what are the various functions we have seen about the plasma membrane, then cytoplasm, endoplasmic reticulum, mitochondria, ribosome, lysosomes, Golgi apparatus, nucleus, as well as a nucleolus. So this is about the brief, the summary of the structure of an animal cell. So today, we have completed the structure of an animal cell, that is ultrastructure of an animal cell. So don't get confused. Structure of an animal cell, ultrastructure, why we are calling it as an ultrastructure is because in the ultrastructure you can observe minute details, and this is possible only with the electron microscope, not with the ordinary microscope. So if you look at the structure of a cell, that is observe very keenly using an electron microscope, there since the electron microscope has got high resolving power and also high magnification, even the minute details, that is the ultra details of a cell can be observed very, very cleanly. So based on this electron microscope study only, the scientists actually have given us the structure of a animal cell, not only animal cell, plant cell, or human cell, whatever it is. So they have given in depth. I mean, very keenly they have observed and they have given us in depth the minute details of a animal cell. So that is the reason why today very authentically we are able to say that the animal cell, not only animal cell, that is in general the cell actually is bounded by means of a nuclear, plasma membrane, and beneath this plasma membrane you have what? You have got the protoplasm, which is basically differentiated into cytoplasm as well as nucleus. Cytoplasm has what? The organelles as well as the paraplasma. And the center one, you have got the nucleus, which is bounded by nuclear membrane, and the nucleus, you have got the nucleolus, nucleoplasm, and you have got the genetic metal in the form of protein network along with the protein as well as the ribosomes. So that is all about the ultra structure of an animal cell. And each, what do you say, the structure of a cell is very, very important. And each cell actually has got, what do you say, the role to play, because since cell is considered to be the basic as well as the structural and as well as the functional unit of a cell not only gives support and structure to the body, I mean, to the, what do you say, the tissue, but it also actually helps or brings about the various metabolic activities which are essential for the, what do you say, the metabolic activities, which are essential for the various metabolic activities. Because without the cell, there is no life at all. So that is the reason why cell is also considered as a unit of life, and cell is a, what is a place where all the different metabolic activities that are basically required for the existence of life, whether it is an animal or whether it is a plant or whether it is a human, is very, very essential. So with this, today we are going to, close. In the next class, we will discuss about the various, what do you say, the chapters.